Good afternoon, everyone. I'm Nikki Kinloch. I'm the CEO of Cure Cancer. Thank you for taking the time to join us at our very first Lunch and Learn of 2024. It's a pleasure to have Dr Adel Aref here, who's based at the Children's Medical Research Institute, or CMRI, known to some of us, in Sydney in New South Wales. I'd also like to extend my gratitude to the Julius Shirodi family and friends whose generous contributions have enabled us to fund Adel's novel project on mesothelioma this year. Thank you so much. This session will last around 45 minutes, and if you do have any questions, please type them into the chat box. You'll see that below or on your right, depending on your setup, and I will weave those into the conversations with Dr. Aref as we go along. Okay, if you do have anything else that you'd like to ask and you don't have the opportunity, you can always email us after the session. So that's info@curecancer.com.au and we'll come back to you on your question. So it's, as I said, a delight to have our first session this year. Thank you for taking the time. Dr. Adel is a clinical and medical oncologist with over 10 years of experience working in Egypt, the UAE in Dubai particularly, and the Kingdom of Bahrain. Although he found clinical practice rewarding and fulfilling, his passion for cancer research never waned. With his Cure Cancer funded project, Adel aims to identify a molecular signature, if you like, that would predict the likelihood of a response to a treatment known as immunotherapy in patients that have advanced malignant pleuromesothelioma, or MPM. Whilst MPM is rare, Australia has the highest number of cases worldwide, and we'll dig into that shortly. This is a serious disease and is often caused by exposure to asbestos. Symptoms usually don't show until the disease is advanced, and that leads sadly to late diagnosis. Unfortunately, only around 10% of patients survive more than 5 years after their diagnosis of MPM. Next week, on February the 29th, which is Thursday, it marks a Global Awareness Day for Rare Diseases. And in light of this, we're here with Dr. Adel to answer your questions and learn more about his groundbreaking research on mesothelioma. Adel, welcome. First of all, could we just start by talking a bit about you? For those that don't know, do you want to introduce yourself and give them a bit of your background?
Sure. Thank you, Nikki. It's a great pleasure to be here today with you and with Cure Cancer. So, my background is, as you mentioned, I'm a medical oncologist from Egypt. I finished my specialty training 2008, which is a long time now. Then I worked as an oncology specialist for 1 year in Egypt. Then I moved to Dubai. I worked there for 5 years as oncology specialist, and I finished my specialty certification from the European Society of Medical Oncology and from the Royal College of Physicians in specialty certification medical oncology. My interest in research was building up, so I attended few workshops in cancer research and clinical trial methodology. I moved to Bahrain in 2015. I worked there for 1 year. Then by that time, it was clear to me I want to continue in cancer research. So I applied for a PhD, and I was lucky. I was awarded a PhD scholarship from the University of Adelaide. I moved to Adelaide in 2016, mid of 2016. Started my PhD. It was about prostate cancer and the factors affecting prostate cancer screening and why some people have more aggressive prostate cancer than others. I finished my PhD in 2019. As part of my interest in cancer clinical trials, I worked in School of Public Health as part of the Health Technology Assessment Group. So I was reviewing clinical trials submitted or applications submitted for listing new drugs in the PBS system and Medicare system. I worked there for 1.5 years, 2 years. And then I was still eager for more research. So I joined CMRI in mid of 2021. Um, I'm working as a biological lead for several projects, mainly aiming to develop biomarkers to predict patient prognosis or their outcome, if they will be doing well or doing bad over the course of their disease, and to develop also biomarkers to predict whether they will respond to specific treatment or no. Part of
So
this
Adele,
is
sorry, could I just say you're talking about biomarkers there. Could you just explain to people that may not know what is a biomarker?
sure. Biomarker is um, a test
Mhm.
um, that can be based on tissue or blood, and it reflects the aggressiveness of the disease in very simple words. So it can tell you whether this disease will be aggressive or there is higher risk that the disease will progress rapidly or vice versa. The biomarker can be based on a blood sample, protein in the blood, or protein or gene in the tumor tissue. So what we are doing is trying to find this marker which is based on protein
Mhm.
in the tissue at time of diagnosis, so the baseline tissue when patient got diagnosed with a type of cancer. So as part of the diagnosis, we take part of this tissue to know what type of cancer, what is the characteristics of this cancer. So we are also analyzing these tissue proteins to identify uh, a marker of few proteins that can tell us whether this tumor or this patient will respond to a specific treatment or will have a good prognosis or bad prognosis.
Okay, thank you. So you obviously you started off in one cancer type, and now you've moved across to more of an immunotherapy focus. Could you just explain in your words what immunotherapy? I mean, everyone probably has heard of this now, but just want to recap: what is immunotherapy, and why is that so important nowadays?
Okay. Immunotherapy. So, one of the reasons why cancer is very hard to treat is that they mask themselves from our body immune system. So our bodies have the capability to identify any foreign object or foreign tissue and attack it so that it destroy it without affecting the whole body and without making it cause any harm. Um, Cancer across its different types have the ability to mask itself from the body immune system. So what immunotherapy do simply is that it um, close this mechanism, the cancer ability to hide itself from the immune system and enhance the body immune system to attack it and destroy it or make it shrink. Um, it is important because it's more or less a common mechanism across different types of cancers. So it has shown efficacy across different types of cancer. And because it's more or less more specific than the conventional chemotherapy, so overall the scope of side effect is much better. Immunotherapy is compared to chemotherapy is more tolerable. And in some disease area, it's showing very good response rates.
Lovely, thank you. So I just want to go back to your focus on MPM or advanced malignant pleural mesothelioma. Could you just maybe take a step back and I mean, I think most people in Australia would be aware of it or have heard the word mesothelioma, and as we know, sadly, we have the highest number of cases worldwide. Could you just explain what that is and then what the difference between that mesothelioma and advanced pleural mesothelioma is, please?
Okay. Um, Mesothelioma is a type of cancers that arise from the thin layer, which we call the pleura, which cover our lung. So it does not arise from the body of the lung or the lung parenchyma. It arises just from this very thin pleural or thin layer. It's directly related to exposure to asbestos. And like in areas where there was, especially during the last 30, 40 years, when there was higher exposure to asbestos because of industrial activities and whatever, the rate of mesothelioma is much higher than other areas where there is like agricultural or there isn't much industry, heavy industry there. Advanced malignant mesothelioma is the same disease, but when it is diagnosed or at an advanced stage. So advanced stage is when excision or removal of the tumor is either cannot be done completely or impossible because the tumor is big and it's like attached to everything around it. So this is what we mean by advanced malignant mesothelioma. So it is the same disease.
Yeah.
It is just the advanced stage of it. As you mentioned, mesothelioma, despite being a rare disease, it's a very aggressive disease. The average survival is around 1 year, and less than 10% of the patient can survive 5 years. The research in mesothelioma, although it has been going on for a long time, there was no great steps and advances during the last 20 years. So before 2020, when the combination of immunotherapy was approved, the last previous approval was maybe 15 years before this, when pemetrexed and bevacizumab were approved for use of treating mesothelioma. So yeah, it's the entrance of immunotherapy in this disease area was of great interest. Like people was having a lot of hopes, but yeah, it's tricky.
Yeah, absolutely. I mean, they're really sobering numbers that, whose survival rate over 5 years is less than 10%. I mean, that's staggering. So, thank you for looking on at this piece of research for us and making advances here. So like many types of cancer, chemotherapy was the traditional treatment option for this, and now you have the immunotherapy coming in as well. So when you're looking at your research, I understand obviously you need samples. So how do you gain your samples to enable you to do your research and be able to test?
Yeah, it's a important step. Actually, it's a fundamental step. So it's usually based on research, networking, and collaboration to find the research centers or groups or biobanks which have tissue with clinical data, because availability of tissue alone is not enough. We need clinical data and we need follow-up data to be able to develop the biomarker and do the research. So based on like searching, networking, and knowing what is going on in this disease area, we start exposing uh, opportunities for collaboration. And when we success, we can identify a group, a research group, or a biobank who are interested in our research, in using our technology, and they have the resources, the tissues, the clinical data to integrate it in our research, and so that we can build this research and answer the important research question.
Thank you. So does that mean that you're working with international collaborations as well, or is it just based in Australia?
No, it's both national or Australian and international collaboration. So, for example, this project, it includes two cohorts, one from the Australian Asbestos Institute, the ADRI, and the other one is from the International Mesothelioma Network, which is located in France. And it is one of the biggest international research network in mesothelioma. They are involved in most, if not all, the important phase 3 clinical trials that involving mesothelioma and treatment of mesothelioma, previously in the era of chemotherapy and currently in the era of immunotherapy. So this project is built on a national and international collaboration to get samples from both from here, from Australia and from France, for patients with advanced malignant mesothelioma treated with immunotherapy, either as a first line or as a subsequent line, like when progress. And we will use our technology to identify the biomarker I was talking about to see if we can better select patients who have higher chance of responding to immunotherapy. We will also be able to identify possible new therapeutic options for patients who are not responding to immunotherapy, which overall, so like the overall outcome of our research is to improve patient outcome, whether they are good responders to immunotherapy, or even if they are not good responders to immunotherapy, we'll be able to find alternative options, which theoretically should improve everyone's outcome.
Thank you. I'm just going to pause there and just welcome, we've got a couple of new members. So welcome to this session, the Lunch and Learn. It's a pleasure to be talking to Dr. Adel Aref at CMRI in Sydney, and we're talking about mesothelioma cancer research. Before we carry on talking about the biomarkers and the work you're doing, I do have a question here, and if anyone else has a question, please pop those into the chat and I will weave them into the conversation. One of the questions, Adele, was can you be predisposed or is it genetic, or is it only environmental factors like asbestos that can cause mesothelioma?
Okay, it's a tricky question. It's mainly environmental, but there may be some genetic factor, but it won't be. So, okay, let's say it that way. For every 100 person exposed to asbestos, around 10 to 15 will develop mesothelioma. Okay. But among 100 patients with mesothelioma, 90% or more have been exposed to asbestos. So the main factor is environmental exposure, but for sure there is other genetic variations which make this exposure lead to development of cancer, or it can be more protective. So it needs more exposure, or it can even protect from the side effect or from the effect of asbestos. Not sure if this is clear.
Yeah, no, that makes sense. So yes, there is the possibility that it is more than asbestos, but in 90% of the cases, it's exposure to asbestos.
Exactly.
Thank you for answering that. Lovely. Thank you. So you're acquiring these samples through international collaborations. You are looking at, obviously, traditionally it was chemotherapy, and now we're looking at those patients that are most likely to respond to immunotherapy. Now, how do you treat a person with mesothelioma with the immunotherapy? Could you talk about what that treatment plan looks like? Obviously, everyone's plan is slightly different, but how does that work?
Sure. So when we are talking about treating patients, first it's whether patient is treated in the context of a clinical trial, which can take a lot of forms and variations, and there is a lot of clinical trials going on for immunotherapy with different agents, either as a single agent immunotherapy or combination with or without chemotherapy. And the other context is treatment in public hospitals based on the approved regimens. So currently, the only approved um, immunotherapy regimen for malignant mesothelioma is a combination of nivolumab and ipilimumab, which we call nivo-ipi. This is based on a clinical trial which was published 2021. It's called Checkmate 743. And this clinical trial shows that this combination of immunotherapy compared to chemotherapy lead to better um, survival, which was around 2 to 3 months survival benefit. Like patient lived on average 2 or 3 months more, and the response rate with the combination immunotherapy was around 40% compared to around 15 or 20 in the chemotherapy arm. So this is the currently approved regimen for immunotherapy. Other regimens include, as I mentioned, different types of immunotherapy, a single agent with or without chemotherapy, but none of them is yet showing enough strong results to make them an approved treatment within the healthcare system, like Medicare or others.
Okay. I mean, even just when you're talking there, it's shocking that even after that clinical trial, although there's been success, it's only extending patients' life by 2 to 3 months, which is still a bad result. Obviously as a patient, it's better, but it's clear that so much more work is needed to be done on this research, isn't it? So
Exactly.
yeah, what's your hope then? So you're looking at these biomarkers, you're looking at patients' potential responses to treatment plans through your research that you're doing now, which is being generously funded by the Julius Shirodi family. How are you seeing this turn out? What are you hoping for to find and discover in your research?
Sure. So our hope is to develop what we can call patient selection tool. So at time of diagnosis, we do a small test on the tissue already taken for diagnosis. And based on a few number of proteins, we can say, okay, this patient will most likely respond to immunotherapy. So by this, we know that the chance of responding will not be 40%. It will be much more than this. It will be 80 or 90%. So this means that we are giving a treatment to a patient that we are more confident it will be working. So even if there is side effects, the overall benefit will be better because we know there will be response. We will also be avoiding patients who we know that they will not be responding to immunotherapy from giving them treatment that it is not working, it's expensive, and it will have a side effect. And it will just waste their time taking something not working unless we know this upfront. So this is the ultimate goal of the project, to select patients early and give immunotherapy to patients who we are more confident that they will respond to immunotherapy. So this on patient level, on physician level, on the public health level, it's a win-win for everyone.
Absolutely, absolutely. And given the numbers are so poor at the moment, having that success would be wonderful. So could you talk through a bit more of the timing or the process? So you're doing this research now. You get some positive indicators that your research is working. You've identified biomarkers. What happens after that? What are the next steps from ultimately where you are to one day having this as a treatment for people that have got mesothelioma?
Sure. To make this biomarker available for patient in the clinic, it need to be we need to show it is working. We need to confirm it is working, and we need to make the regulatory bodies believe that it is working so that it can be used in clinical practice. To confirm it is working and make sure that the regulatory bodies are confident that it is working, it's what we call validation. So, and this is one of the reasons why we try to get two cohorts from two different places, because it's easy to identify a biomarker within a cohort of patients and show it is working. The difficult step is to show it is working in another completely different independent cohort of patients. So this project, with its current aims, will we will develop or build a biomarker in one cohort and hopefully show that it is working also in another independent cohort. The next step after this is to confirm it in a bigger third or more, like one or more cohorts. And based on the data, how strong it is, we will have enough information to start the process of getting it approved by regulatory bodies like the TGA. There will be enough evidence to use this biomarker in future clinical trials. And this, at this stage one, it is approved at least on the TGA level and start to be used in clinical trials. This is when patient will start getting benefit from it because patient will be using it for selecting their treatment.
Thank you. That absolutely, that makes sense. Just a question around the cohorts. You know, you talked about how you've got samples from different countries. Is MPM the same for people that have MPM in one country versus another? Is it, or do you have different variations of MPM?
So MPM is a heterogeneous disease by default. So there is three big pathological subtypes: epithelioid, sarcomatoid, and non-epithelioid, or a combination. So, and it can be classified as epithelioid and non-epithelioid. This is not based on ethnicity or a regional classification. It's a pathological classification. Because as we mentioned, for sure there is a genetic variation and there is a molecular heterogeneity within mesothelioma. So if we have enough number of samples from different countries worldwide with different ethnicity backgrounds, I'm sure we will find that it can be classified in completely different way. But currently, like, the assumption is if you have one patient from Australia, one patient from France, both of them having non-epithelioid mesothelioma, they are more or less the same. Like, let's say 90% the same.
Yeah. Okay. Thank you. That makes sense. So you talked very much about where we should be going from your research to clinical trials. You know, I don't know if you can even answer this question, but how long do you think that would take? Is that a 5-year, 10-year? What are your hopes from a traction perspective?
My hope is to finish it ASAP. Reality, it can take time, which will be years, but it depends on how strong is the data and based on the strength of the result, how long it will attract future collaboration to validate it and to what extent it or how quickly it will be approved by the regulatory, at least on the level of the TGA and the FDA. Still, we are in the first step, but we are, like, let's say we are combining the first and second step. We still have another 3 or 4 steps. And if we had to use or include this in a prospective clinical trial, it may take another 3 or 4 years. So yeah, it's still the early steps, but we are hoping with strong results that we can move things quickly. And hopefully, yeah, patient can start using it even in the context of a clinical trial within the coming, let's say, 4 or 5 years.
Thank you. Yes, it's interesting. We last year I was talking to we funded a couple of lung cancer researchers last year, and they particularly were saying that, and I don't know if it's the same for the mesothelioma research, but for lung cancer research, it gets to clinical trial much quicker than other types of cancer, purely because there's been very few advancements in lung cancer research in the last 30 years. So that, you know, lung cancer is sadly the biggest killer in the world for cancer. So I'm hopeful that we'll have the same situation for you in mesothelioma.
Yeah.
Thank you. Okay, one question here, interesting, about the age profile of the patients with mesothelioma. So obviously asbestos in houses was known in the 80s, particularly here in Australia. Are you dealing with an older generation of patients now because that was a bigger issue then, or are you still seeing younger or having samples from younger patients because of building work, etc., where there's asbestos as well?
Sure. The majority of our cohorts are not elderly, but towards elderly age, so not young age. Think so. This is there is a lot of variation in the age at time of diagnosis for mesothelioma. So in most of Western countries, it's usually 50s plus. So it's related to more or less minimal exposure for long time. But in other areas where there is very high exposure within either like people living in areas where is factories like very adjacent to factories, or workers working with in industry with very high exposure to asbestos and without enough protective measures, mesothelioma can be presented in very young age, like 20s and 30s. So this have been seen, and it's a balance between exposure duration and protective measures. So yeah, as an answer of the question, most of our cohorts are towards like, let's say, around 50 plus. But this doesn't mean that mesothelioma cannot affect young patient, or young people, but it's within a different environmental exposure context.
Understand. Thank you. Another question just on that. So with asbestos being banned now in Australia, are you seeing your samples from the Australian cohort reducing? You would presume less people with mesothelioma now in Australia versus other countries?
A number of patients is relative to the whole population.
Yep.
So like 1% of a million will be much less than 1% of 100 million, for example. But the incidence, so the number of new cases of mesothelioma in Australia is expected to keep increasing for, I think, the next 4 or 5 years to peak, based on all the previous exposure during the last 20 or 30 years. So all the previous exposure and before getting asbestos banned and have all the measures in place, the effect is not yet seen. It will take another few years before we start seeing decrease in incidence of mesothelioma.
Okay, thank you. So there's a bit of a lag effect there,
Yep.
unfortunately. Okay, thank you. A question really now about technology. We all talk about it, and we talk about AI, artificial intelligence, and statisticians as well. Do you use any of that in your research?
It is a fundamental part of our research. So when I'm saying we are like identifying a number of proteins that are associated with response or prognosis or outcome, this is all based on very sophisticated and complicated machine learning modeling, and combining it with conventional statistics to find these proteins, because the number of proteins is huge. You're talking on average of around 4,000 to 5,000 protein per sample. and we need to search within these 5,000 protein, for example, to find only 5 or 10 maximum that are working together and can be used as a biomarker. So uh, the whole analysis depend mainly on machine learning and combining it with statistical modeling, regression modeling, and survival analysis to find these proteins and show its association with outcome of interest, whether survival or response.
Thank you. Okay, makes sense. And then just a question, you talk about looking at the proteins. Why specifically are you looking at the proteins for this analysis?
It's not for this analysis, it's our whole program, it's our whole research pipeline that we are like during the last 20 years, it was the era of genomics. So genes and the mutation in genes or how the changes in the genes can affect cause disease or be associated with response or um, outcome. But like genes is not the final level. We still have proteins, which is the end product of these changes in the gene. So when you have a mutation in a gene, it's it is not what is causing the disease. It is how this is translated into change in the proteins, which is the building unit of every cell and every interaction, every function happening in the body. So all the function or most of the function are happening on the protein level. So this is the idea of our whole program, is that to do the analysis on the protein level because it's more representing. It is actually what uh, treatment, chemotherapy and targeted chemotherapy and targeted treatment and immunotherapy um, target at the last stage. It's all about proteins. So this is why we believe that using proteins will give a more robust result. Biomarker will be more representing, and if we identify new targets at this level, it will be more promising and more useful to use.
Thank you. Just like to remind the audience, we've got about 5 minutes, so I'm just going to have a couple more questions for Adel, but if you had anything else you wanted to ask, please drop it into the chat box now. So Adel, this funding obviously has come at a good time for you. Can you just talk about the importance of funding like this, and the challenges as a researcher? And I know I know very well, but for our audience, what are the challenges for researchers in getting funding?
This is the biggest challenge, especially for early career researcher who are starting their research career. It's very difficult, it's very competitive, and as I said, the hardest is the first one. So yeah, this funding was a great opportunity to and it it is a main factor that will allow this research to continue and to have results. It was a great opportunity for me to build my career as a cancer researcher and proteomic researcher in the biomarker discovery. So yeah, it is a main problem that can just hold back the whole research, and like without funding you cannot do any of the things we are doing. So yeah, it's critically important. Funding early research early career researcher is critically important because the best ideas and the most innovative ideas are usually from people who are starting their career, but like getting funding at this stage is very critical. And yeah, one of the things that I really um, uh, like and love in cure cancer is that they are targeting mainly early career researcher, because this is where most of the innovation, but most of the hectic to get to find support and acknowledgement of what we're doing.
Yeah, no, absolutely. It's part of the ethos of what we do is we try and kickstart careers or brilliant new ideas that, you know, as you say, a wealth of them come from early career researchers, and unfortunately it's very difficult for ECRs to get funding as well. Just one question, I don't even know if I'm pronouncing this correctly. Can we use phage therapy in mesothelioma treatment? Phage, H-A-G-E?
Not sure I'm getting this
No?
correctly. No.
No, okay. Well, maybe the person that typed that could just clarify what they meant there, and we can revisit that question. So you talked very much about collaborations as well, and you know, for you, obviously there's a big focus of it coming out of France too. What's your hope once we've gone to clinical trials? That will make, I presume, the life of a clinician easier, because it will give them multiple different tools to be able to impact treatment,
Sure.
patient treatment as well. Is that correct?
Yeah, of course. It's like when we reach the clinical trial stage, this is giving more treatment options or even selection options to the clinician. It will make patients more confident that they are receiving the best treatment for them. And this is the whole idea of precision medicine. It's like you have a lot of treatment options, but you try to make your best to give the right treatment to the right person. So yeah, hopefully it will make everyone's life better, including the government, by saving money. So yeah, it is the ultimate goal. It is the end product of our research.
Absolutely. Well, thank you, Adele, for your time today. Very much appreciate it. Thank you to you and your team at CMRI for the incredible work you're doing. We're dealing with a cancer that sadly at the moment only has a 10% survival rate over 5 years. Last clinical trials saw a 2 to 3 month improvement or length of improvement of a patient's life. So we've still got a long way to go. So your research is incredibly vital on that. So thank you from all of us and on behalf of the audience for the work you're doing. And I wish you every success in that as well.
Thank you so much. It's a pleasure to be with you.
Thank you.
Thank
And
you.
thank you to our audience as well. Please join us at the same time on Thursday, the 14th of March for our next Lunch and Learn. That's with Dr. Emily Gruber, and she's at the Peter MacCallum Cancer Centre. Her focus is on blood cancer, particularly acute myeloid leukemia. So if you can, please join us then. If not, like anything, these conversations will be sent to you next week, so you can review them at your own time. But in the meantime, thank you, Adele. You've got lots of fabulous feedback here in the comments section. Thank you. Well done. And thank you to all of you on this call for helping to support brilliant young researchers such as Adele. Thank you all, and have a lovely Thursday afternoon.
Thank you.